Okay, so this is chapter 15, part 4. And in this part of the chapter, we're going to focus on the genetic code. Again, I'm going to show you this again before we get into the actual process of translation. We'll also briefly talk about proteins, and we'll talk about tRNA. That'll be our focus for this part of the chapter. So let's jump in. And I'm going to start off by talking about proteins. Okay, so again, our DNA is transcribed into RNA, and then our RNA is translated into proteins. And so before we talk about that process, I want to just briefly mention proteins again. And so we talked about proteins a couple of different times this semester. We really saw them in chapter 3, so you got to kind of remember back to those. We talked about what proteins are made of and how they're formed. So let's quickly review that before we get into the process of translation. And so again, genes are eventually translated into proteins. So RNA is kind of the middleman. What are proteins? They're the functional units of cells. They give you, your cells and you, your phenotype. Okay, they are macromolecules. They're large, complex molecules, and they're made up of the monomeric subunits called amino acids. And remember, there's 20 naturally occurring amino acids. In the beginning of this chapter, I showed you the 20 different amino acids. Okay, so hopefully you can kind of picture that in your mind. You don't need to know structures. You just need to know that's what makes up proteins as amino acids. There's 20 naturally occurring. And importantly, they have differing side chains. And so we'll talk about that again here. They vary in length, right? So some proteins could be 50 amino acids long. Some proteins could be over 1,000 amino acids long. So they're going to vary in length. I'm showing you here in this picture the structure of 2 amino acids and how they are joined together. So let's look at that together really quick. So this is amino acid number 1. So here is the central, what we call asymmetric carbon. It's bonded to an amine group, which is an NH2, and a carboxyl group here, and a hydrogen. What's here, this R, remember, is going to be different depending on the amino acid that we're looking at. If this is just an H, that would be glycine. If it was a methyl group, a CH3, that would be alanine, and then it gets more complex from there. Remember, some side chains are nonpolar and hydrophobic. Some are polar and hydrophilic. Some are charged and hydrophilic. Some have ring structures. Okay, so again, what's here makes the 20 amino acids different from each other. Here's my second amino acid. It has the same backbone. Again, here's the R group, which will be different. To put these two together, that's what happens during translation. So this thing called the ribosome is going to read the mRNA, one codon at a time. A codon is 3 bases long. And as it reads those codons, it's going to bring the appropriate amino acid in to the complex, and it's going to catalyze a peptide bond formation. Okay, so that's here. So again, we have 2 amino acids. They're being brought together. Water will be removed, so here's H2O, and a bond will be formed between carbon, the carbon of the carboxyl group of the first amino acid, and the nitrogen of the amino group of the second amino acid. So in this bond right here is called a peptide bond. Okay, so that's how amino acids are put together. And again, this will be catalyzed by the ribosome, and they're going to be put together in a specific order according to the mRNA, which was made according to the gene. So again, the sequence of the bases of the gene specify the sequence of the bases in the protein. We're going to simplify the process. However, I do need you to know that there are over 100 different kinds of macromolecules involved in this process. That includes proteins, rRNA, ribosomes, mRNA, tRNA carrying amino acids. Okay, so again, we're going to talk about the basics, but I need you to understand that the process is more complex than we're making it out to be. Okay, I want to remind you of this. We saw this once already at the beginning of the chapter. When we talk about translation, again, we're using information that originally came from our DNA that was transcribed into our mRNA. And that mRNA, the sequence of the bases, is going to tell us the sequence of the amino acids in our protein. And it's based on the genetic code. And so that's what this is. Okay, so this is showing the sequence of the bases in our mRNA and what amino acids are then made. So it's really important to remember that your mRNA is going to be read 3 bases at a time. And so we call it a triplet code, and we call each a codon. Okay, so 3 bases of mRNA is 1 codon, and that will specify a specific amino acid. Okay, so let's look at this again. So let's say in our mRNA, the first base in our triplet codon is a C. So we're in this box here, in this row. Let's say the second base is an A. We'll come over here to this box. So that brings us right here. We're here. And let's say the third letter in our triplet code is a G. So that brings us to the bottom. So again, CAG. That is going to code for this amino acid here. Okay. I want to just point out to you start and stop codons again. And I know I mentioned this back, in the beginning of the chapter, but just to make sure we have it. Where's my pointer? So here, AUG, this triplet codon, is going to be the start codon. It's the universal start codon, no matter if we're looking at prokaryotes or eukaryotes. And it's going to code for methionine. Okay? In prokaryotes, it's actually going to code for FMET, which is formylated methionine. We're going to talk about that in the next part of the chapter. In eukaryotes, it's just plain old methionine. Stop codon will be at the end of the gene. And so UAA, UAG, and UGA, all 3 of these code for stop. So when the ribosome hits one of those codons, it's told, okay, we're at the end, stop. Okay? And we'll talk about how that happens in the next part of the chapter. Remember that the genetic code is universal, and it's also redundant. Okay, we covered that back in the first part of the chapter. Okay, so that's a reminder of our genetic code. So the last thing I want to talk about is tRNA. And so, again, mRNA, the M stands for messenger, and that's what's actually going to be read by the ribosome during translation. And rRNA is part of the ribosome. It has enzymatic functions. And lastly, tRNA, the T stands for transfer. And so, again, that's where I want to focus for the rest of this part of the chapter. So what happens is amino acids are going to be covalently linked to these tRNA molecules by an enzyme called the amino acyl tRNA synthetase. And what you end up with after you have an amino acid attached to your tRNA is an amino acyl tRNA. Okay? This connecting of an amino acid to the tRNA is essential to the function of tRNA. Its job is to bring the right amino acid to the ribosome during translation. How does it know what the right amino acid is? So this enzyme that catalyzes this reaction, it's actually going to look at the bottom of the tRNA molecule. There's going to be a triplet of bases, so 3 bases, which we are going to call the anticodon. And depending on what that anticodon is, that's going to tell us what amino acid to add. The anticodon is going to be complementary to the codon in the mRNA. And again, it's the enzyme, the amino acyl tRNA synthetase, that sees this, recognizes it, and adds the right amino acid. So let's take a look. I got a couple different figures to show you. They show this in different ways, and I kind of like all of them, so I decided to include them all here. So let's start off on the left. And so this is what a tRNA molecule looks like, and I told you back in the beginning of this chapter that tRNA is single-stranded. It has a 5' and a 3' end. It has a sugar phosphate backbone. It has the bases A, C, G, and U. And I also said for tRNA that it kind of folds back on itself and forms a T-like structure. And that's what this is. Okay, so notice there is these little dotted lines in between some of these bases represent hydrogen bonding. And so there is this folding of the molecule. Here is the 5' end. Okay, if we go down the molecule here, there's this region here called loop 1. We go down, we've got loop 2. Go up the side, loop 3, and then up to the 3' end. Okay, and so it is on the 3' end of the molecule where the amino acid will be attached. And it's down here at loop 2, at the base of this molecule, where we have what is called the anticodon. So this is reading AAG. And so let me back up a second. Okay, so AAG, if that's the anticodon, that is going to bind to UUC, which is right here. Okay, so AAG, if that's the anticodon, the codon, its complement, would be UUC. Okay, and that codes for phenylalanine. Okay, so again, the amino acid tRNA synthetase looks at the anticodon, and based on that anticodon, it puts on the right amino acid. Okay, let's go back here. Okay, this is the same exact anticodon here, just drawn a little bit differently. So again, 5' end, this would be loop 2 down at the base, 3' end, and here's my amino acid, and look, it's phenylalanine, right? So the anticodon AAG has to be complementary to the codon UUC. And so again, if you go back, flip back in your notes, and look at the genetic code, again, UUC codes for phenylalanine. Okay, so we got the right amino acid on there. So what's going to happen during translation is as the mRNA is being read, tRNA will come in and it's going to have to bind to the mRNA in a complementary fashion. And so down here, the anticodon will be complementary to the codon in our mRNA. And so that's what ensures that the right amino acid is brought in. Okay, so the codon is in the mRNA, the anticodon is in the tRNA. And again, that ensures the proper order of amino acids as we make our protein. So I just wanted to show you this. It does take energy, and the enzyme involved here is called the amino acid tRNA synthetase. And so again, it sees the anticodon, it brings in the appropriate amino acid, and attaches it to the 3' end. There's just one other figure that I liked. This is from the textbook that we have as a reference. So it's from the OpenStax biology textbook. And this really shows how the amino acid tRNA synthetase does this. It's pretty neat. So if we look at our tRNA, okay, and this specific tRNA has the anticodon ACA, okay, this will bind into this active site on our amino acid tRNA synthetase. The enzyme here sees the anticodon ACA, and it's going to bring in the appropriate amino acid. And so the complementary codon to this in our mRNA is going to be UGU. So flip back in your notes to the slide on the genetic code and find UGU. Take a second to do that. Pause me if you need to. You should see that UGU codes for cytosine. And so that's here. This little blue hexagon, right, is cytosine. And so the enzyme will also take up cytosine. It will use ATP, and it will attach the cytosine to the 3' end of our tRNA. And then this will be released, and it can go ahead and it can do its job. Okay, so again, this part of the chapter was really to introduce you to tRNA and to describe to you how the right amino acid is attached to these molecules based on the genetic code. Remember, anticodon will be in our tRNA, codon is in our mRNA, and those have to complementary base pair during translation. Okay, and remember again, it's the tRNA's job to bring in the right amino acid. Okay, in the next part of the chapter, we'll get into the nitty-gritty of translation.